All right, chapter 11 is a continuation of chapter 10, but now instead of just going over muscles, we're going to actually go into physiology of muscle. And we're going to use to discuss this skeletal muscle in particular. And then at the end, we'll discuss a little bit more about how smooth and cardiac muscle varies from this. And in contrast, how they compare to each other as well. All right. So here are your learning outcomes as usual, but again, focus on the study guide. Okay, so when we're talking about our muscle cells, so again, remember with this, we're talking about a muscle fiber or muscle cell, referring to, in this case, for some of this, the skeletal muscle cells. So that's what you see pictured here. You can see that we've got really, really long cells. You don't even see the whole thing. They've got more than one nucleus, and they're striated, right? They got those lines on there. So we're going to go over what that is. Now, when we're talking about skeletal muscle, or when you're talking about muscle cells, the sarcolemma is the term that's given to the cell membrane, or the plasma membrane. It's called the sarcolemma. So just remember, when you see myo or sarco, it refers to muscle, all right? The sarcoplasm then would be the cytoplasm inside of the muscle fiber. Now, again, this multiple nuclei is referring to the skeletal muscle cells in particular. Now, things that we'll talk about are glycogen and myoglobin. So glycogen, if you remember, is the way that animals store sugar. So those long, long chains of glucose molecules that are branched, that's called glycogen. And that's often stored up in the liver, but also in muscle. And then we have this molecule called myoglobin. And myoglobin is very much like hemoglobin, but it's found within the muscle cell and it's holding on to oxygen. So as you imagine, muscle cells need a lot of oxygen to make ATP. So they have their own store of myoglobin and actually of oxygen on myoglobin. And actually, oxygen has a higher affinity, or it likes to bind myoglobin more than it likes to bind hemoglobin. And that is sort of the incentive for it to leave the bloodstream and enter the muscle cells. So muscles can store up their own storage of oxygen using myoglobin. And then there are some outside cells called satellite cells, which are unspecialized, we say unspecialized myoblasts. Remember the myoblasts from the last chapter were fused together to make muscle cells. They would remain in between there, and they can help repair or regenerate to a certain extent, these muscle cells. Now, we had left off looking at what's happening outside of the cell, right? We talked about the fascicles, which are bundles of muscle cells, our muscle fibers, and we said that the fascicles were surrounded by the perimysium, that connective tissue, and the muscle cells themselves are surrounded by connective tissue called the endomysium. And then remember, the whole muscle belly is surrounded by the epimysium, all right? And then I said to you, okay, then we're going to go into the cell, which is what we're going to do now. Now, inside the cell, so inside the, within here, the sarcolemma here, there's these bundles and bundles of, or many bundles of these fibers that are called myofibrils. You can see they're really stacked up inside the cell. And this is what's causing these striations that you see on the cells, right? So these lines here, because these myofibrils are made up of two proteins, thick filaments and thin filaments that are arranged in a repeating structure called the sarcomere. And the sarcomere is the contractile unit that you see. So these bands here are actually sarcomeres, all right? So I'm going to go over how that's put together, and I'm just going to go grab my notes off my scanner here. Because I started recording the video before I went and got my notes, because I just scanned them in. All right, so what I want you to do is have a look at page 1 if you're following in the notes, and you can see I sort of drew out a sarcomere. All right, so let's go through this. All right, so here you can see the thick and thin filaments. All right, and this would be your sarcomere. Now, and here's another one here, and you can sort of see a whole sarcomere. So the two sides of the sarcomere here, this is called the Z disc, and it holds the thin filaments, which are made up of actin, right here. So thin filaments are made up of actin, and if you remember, that's one of your cytoskeletal components. At that time, I was calling them microfilaments, but here we call them thin filaments in muscle. And the thick filaments, which are shown here in blue, are in the middle of the sarcomere. All right, and they're held there at the M line. All right, so you can see in my drawing on page 1, there's the M line in the middle. Okay, and this is where we are holding our thick filaments here in the middle. All right, so you've got your Z disc on each side holding your thin filaments, your M line in the middle where your myosin is. Notice they're all starting with M. Okay, and then if we look at this in a little bit more detail, all right, so here again, we've got myosin in blue, actin in red. I try to keep it straight like that. Most pictures will do that, but there's some little variations, but most of the time I'm going to do that. Now, here's your Z disc and here's your M line. Now, there's other designations we give these banding patterns. So the whole sarcomere goes from Z disc to Z disc, okay? Now, any place where you only have myosin, because notice that they're overlapping, and that's going to be really important. We'll level that up, but they're overlapping. So any place where you only have myosin, we call the H zone. And anywhere where there's myosin is called the A band, all right? Now, the I band is any place where you only have actin, which you see here, okay? Now, notice that the I band will extend into the next sarcomere, because these sarcomeres, as you can see here, are sort of stacked end to end to end to end within the myofibril, all right? All right, and then here you can see it in 3 dimensions, all right? You can see how these are arranged in these cylindrical kind of structures. And what's going to happen when the muscle contracts is that the sarcomere is going to shorten, and these thick and thin filaments, the actin and the myosin, are going to slide past each other. Okay. Now let's look at a little bit more detail here. All right, there's some detail on the slide. You can see there's this sort of blue thing and this yellow thing here. And here you can see here, this yellow thing is called the T-tubule. And this blue thing, it's not blue really in itself, of course, it's just colorized that way. This thing is called the sarcoplasmic reticulum, all right? So you can see it here. I'm gonna now look at this slide. You can see a little bit more detail, all right? Now here, the T-tubules are purple, all right, here. Now these T-tubules, what these are extensions of the sarcolemma, all right? They're infoldings of that sarcolemma that penetrate deep into the muscle fiber. And here you can sort of see the openings on the outside for these T-tubules. So you can see they're continuous with the sarcolemma, and then they dive deep into the cell, all right? And sometimes they'll even come out on the other side of the cell, all right? Now this blue stuff, here it's also colorized blue. I don't know why, it just happens to do that. This is a structure called the sarcoplasmic reticulum, or the SR. Now, this sarcoplasmic reticulum is actually smooth endoplasmic reticulum, all right? That's specialized for holding calcium. So remember when we talk about sarcoplasmic reticulum as parts of the cell, I said that sometimes the endoplasmic reticulum is part of the cell. I said to you that some ER will act as a storage vessel. Others will help make things like lipid and things like that. But in the muscle cell, that smooth ER the sarcoplasmic reticulum is what it's called here because now it's particular to muscle. It stores calcium, all right? And you can see how it's sort of arranged over these sarcomeres. So here's like a full sarcomere. And you can see that each end of the sarcomere is associated with the T-tubule, pretty much. And then there's this overlaying of the sarcoplasmic reticulum, which contains calcium. And you can see the ends of the part of the sarcoplasmic reticulum that's close to the T-tubule, you can see looks kind of swollen, all right? They're called terminal cisternae or terminal cisterns. A cistern is something that, like a water cistern. If you go to New York City, if you look on the roofs of many of the buildings, there's a big cistern of water, or the water towers, I used to call them when I was living in the city. So those are cisterns. So here, these cisterns are bigger vessels or bigger containers of calcium. And you can see there's two, one from on each side of a T-tubule, and the T-tubule plus two terminal cisternae from two opposite sarcomeres is referred to as a triad. All right, that's what that's referring to here. Okay, now let's look at this differently here. So here you can see there's a T-tubule. Here is a terminal cisternae. Here's another one. Underneath you can see a sarcomere. All right, you've got your thick filaments of myosin, your thin filaments of actin. I'm giving you a little bit more detail now. All right. Now these T-tubules, what they do is they allow for an action potential to penetrate deep into the muscle cell. So what's an action potential? Well, basically, in order for a muscle to contract, it needs to get a stimulus from a neuron, right? And that neuron, as you probably know, that neurons transmit electrical stimuli. And that electrical stimulus is transferred to a muscle fiber or muscle cell using a neurotransmitter. So a muscle cell has receptors for the neurotransmitter. And that then sets up an electrical potential, or an action potential as we call it, across the muscle cell, all right? And that that action potential penetrates deep into the muscle cell itself using these T-tubules. And that then causes the release of calcium from the sarcoplasmic reticulum, all right? So let's think about this in a little bit more detail, all right? So I want to follow along with my notes. There's a lot of ideas I just threw at you here, okay? So if you're looking on page 2, all right, I'm talking about how this works, all right? So slides 12 to 24 are all going to discuss how this works and how the sarcomere is going to respond to that, all right? So we have to go step by step. So let's have a look again at our triad, right? So here we can see our terminal cisternae of our sarcoplasmic reticulum. You can see our T-tubule. And here you can see, I like this one because you can really see the three-dimensionality of this setup, all right? Now imagine then an action potential or an electrical potential spreading across the muscle cell, and it's coming in here. And there's an interaction here. We don't need to talk about that interaction in this course, but if you want to know more about it, I can always tell you a bit more about it. But there's an interaction that happens here, and when the electrical potential comes down, it stimulates the sarcoplasmic reticulum to release calcium. And this calcium then can flood into the cytoplasm around the sarcomere, all right? So clearly something has got to happen here, all right? Because there has to be something different between a contracted muscle and a relaxed muscle. And its key ingredient is calcium, and the other key ingredient, of course, would be ATP, and we'll talk about that. So here you can see, a neuron, and here you can see a part of the axon here, and it ends in this thing called the motor end plate. All right, so the motor end plate, there's a whole bunch of swellings here, and these swellings contain neurotransmitter. So here you can imagine here, you've got this motor neuron. We call it a motor neuron, this particular neuron, because it's interacting with the muscle cell. And it releases its neurotransmitter. The neurotransmitter in this case for muscle, skeletal muscle, is called acetylcholine. So this acetylcholine will drift into the junction here, so the neuromuscular junction or the synapse that we have here, and it will bind to receptors on the muscle cell. This will then set up an electrical potential across the muscle fiber, and it will follow through along the T-tubules, cause a stimulus from, stimulate the sarcoplasmic reticulum to open its calcium channels, and calcium will then flood into the sarcomere. All right. All right. So then with the addition of calcium, all right, we have a relaxed sarcomere and then a contracted sarcomere. All right. So there's, the ingredient here is calcium, and this then allows for this to happen. So when you have a contracted sarcomere, notice that the H zone gets tighter when we do this, but the A band stays the same length, all right? And the other one that will shorten are the I bands. They will also get smaller. And if you look at this using a microscope, you can actually see the difference in the length of the sarcomere. You can see that it actually shortens, all right? This little video here shows you an animation of that, all right? And there's lots of animations of that process. Now, what's the difference now? So there has to be something that's changing with this addition of calcium, all right? So now in this one, now my myosin is no longer blue and my actin is no longer red in this image. So here my myosin is, I guess, tan and my actin is gray, all right? So what I want you to see on these myosin thick filaments is the difference here with a contracted versus a relaxed muscle. Notice that there's these little nubby things that are laying down here. So these little nubbies are laying down in a relaxed muscle, and in a contracted muscle, you can see these little nubbies are sticking up and they're touching the actin. Now, this is because we've got calcium, all right? So calcium is going to allow for this to happen, and I'm going to show you how that works, all right? So these little nubbies here, they're called head groups, all right? And so if we look at these thick filaments with a little bit more detail, all right, you can see that a single myosin is made up of two parts, and the tails will wrap around each other, and then there's two heads. And then there's a whole bunch of these that will intertwine themselves in my myosin thick filaments. All right, it says, according to your textbook, several hundred of these molecules will arrange themselves in a sarcomere to make your thick filament, all right? And then you've got these heads. Now, what's going to happen is that when these head groups interact with the actin, they'll actually walk along the actin, very much like when I was talking about parts of the cell, and I was talking about vesicles being carried around on the microtubules on little feet that sort of walk along there. All right, so these myosin head groups will also walk along another molecule, in this case, actin. All right. So if we look at it, we've got the myosin arranged, attached to the M line, and we've got this, these head groups sticking out. And because they're walking, we have this part that we literally call it a hinge. Now you might imagine that this, of course, is moving, so this requires ATP. And there's actually an enzyme here that's an ATPase that splits the ATP and allows for that energy to do this. All right, the high energy phosphate bond is going to be cut, and the energy is going to be released, so cut by that enzyme. All right, so I'll explain how this works. Okay, now before we get into that, let's look at actin. All right, so now actin, as you know, is also part of your cytoskeleton, but in muscle cells, it's arranged as filaments, so that's your thin filaments. And actin is made up of, or fibrous actin or chains of actin are made up of little globular units of actin molecules, all right? So they're joined together in a chain. And your thin filaments have two of these chains wrapped around each other. Now, in order for myosin to interact with the actin, all right, so the thick filaments to interact with the thin filaments, there has to be a binding site that's revealed on the actin, all right? And in a relaxed muscle, the myosin head groups can't interact with it, But in a contracting muscle, those head groups can interact. And the reason there's a change there is because in a relaxed muscle, those binding sites are covered up by a protein called tropomyosin. And you can see it pictured here as this sort of, grayish protein. And tropomyosin is, onto it is bound another protein or molecule called troponin. Now, troponin is a calcium binding protein. And what happens when the muscle is stimulated and the electrical potential travels down the T-tubule to then release calcium from the sarcoplasmic reticulum? The calcium then binds to troponin, because this is a calcium binding protein, as I said. When calcium binds, that positive charge causes the troponin to change its shape. And when the troponin changes shape, since it's bound to tropomyosin, the tropomyosin also changes shape. And when it does so, it will reveal the binding site, or the active sites on the actin that allow for the binding of myosin. So if we look at actin on its own, the F-actin for our thin filaments, we have these myosin binding sites on here. And when the muscle is relaxed, those binding sites are covered by the troponin-tropomyosin complex. Then when we add calcium, we have a shifting in the shape, or a conformational change, we call it, of troponin, which causes a conformational change or a shape change of tropomyosin. And the tropomyosin then shifts into the gutters of the strings of the G-actin, or the filamentous actin. And that then reveals the myosin binding site. So now the myosin head groups can interact with the actin, and then using ATP proceed to walk along those the actin thin filaments. So here you can see what that might look like. So here you can see your myosin head groups now interacting with the binding site on the actin, because it's now been revealed. So here we can see the difference. We see relaxed muscle, the binding sites covered up by troponin and tropomyosin, which now in this picture are green. And then with the addition of calcium, we can see that the binding sites are revealed, and that then allows for the myosin head groups to interact. And this is called a cross bridge, when the head groups are binding to the actin. So this is called cross bridge. Now, the act of walking along the head groups walking along the actin is called the cross bridge cycle. All right. And normally, if I had a class in real life, I would draw each step individually. All right. But I've got these animations here for you to watch, so you can try and follow through on those. And I'm going to walk you through it. I'm just going to talk you through it, because I'm not going to be able to draw on the board, because we don't have a class together. All right. So now, since it's a cycle, this cross bridge cycle, you can start anywhere in the cycle. All right. But traditionally, one starts here. Now, remember, I said to you that the myosin head groups act as a hinge, right? And I said to you that there's an enzyme component of this that will split ATP. Okay, so keep that in mind as we get going here, all right? So let's go back to my slide with the cross bridge. All right, so I'm gonna start off where we've had ATP bound. And the enzyme then is gonna split that ATP here in step 1, and the ADP and the phosphate will remain bound to the head group. What ends up happening here is, with that energy release from the splitting of that bond, that puts the head group in the cocked position. So it sort of stretches out, all right? So it's cocked and it's ready to do its job. Then what happens is that we lose the phosphate. And when that phosphate is lost, the head group can change shape, reach up and touch the binding site on actin. All right. Now, a quick qualifier here. You can watch certain videos and stuff on the internet, including this one here that I have here, that will say that the loss of this phosphate. So what they'll say is they'll have this, cocked and band, right? So instead of just saying cocked and with ADP and phosphate, they'll say it cocks and it reaches up and touches the binding site. Then the phosphate is released, and then we have tight binding. It doesn't really matter. It depends on the physiologist and which way they look at it for you guys. And to be honest, I don't know what the final consensus is. I do know that we have two different ways of looking at it, all right? But suffice to say, when we're in the second position, the second stage, we have the head groups tightly bound to the actin. So in the end, it probably doesn't matter too much, all right? Now, for the next step, to get to step 3, what's going to happen is that ADP is going to come off. When the ADP comes off, that causes the head group to bend and pull the actin and cause the sarcomere to shorten. Now, of course, we have to do this again, right? We have to do it again and again and again. So we need to release this head group. The way that this is released is that a new ATP will come in and bind the head group, and then it will release, all right? And then we do the whole cycle again. And if you watch the animation, you'll see this repeating over and over and over again, okay? So let's look at it step by step. So step 1, the myosin head groups, the enzyme portion of that has already split the ATP to ADP and phosphate. Step 2 is attachment, when we lose the phosphate and we still have the ADP. Step 3 is the power stroke. So we lose ADP, the head groups bend, and they shorten the sarcomere. Step 4, a new ATP molecule comes in and the head group detaches. Now, as long as we have calcium and we have ATP, this cross-bridge cycle will continue and the muscle will continue to shorten. All right, so let's look at that. So we've got calcium, so we have the action potential, calcium is released, calcium floods into the sarcomere. Calcium binds troponin, changes its shape, causes tropomyosin to change shape. That then reveals the myosin binding site on actin. Then with ATP, the myosin head groups can walk along the actin as we discussed using the cross-bridge cycle. Now, when the muscle relaxes, we're going to then pump. All right, now we have an active pump. We have to actively pump calcium back into the sarcoplasmic reticulum, because when calcium was released, it was diffusion. There's a voltage gated channel here that responds to the voltage, opens the calcium channel, calcium goes down its concentration gradient, floods the sarcomere. Now, in order for the muscle to relax, we have to pump that calcium back into the sarcoplasmic reticulum, and that also requires ATP. Once we lose the calcium, the troponin tropomyosin complex will cover up the binding site for myosin on actin again, and the head groups will not be able to interact. Now, I want you to think about this. This is not something we include in this course. It wasn't in the learning outcomes, but I do talk about it in 105 in human biology. Now, the process of rigor mortis, right? So after death, right? That's the stiffening of the body after death. Now let's think about why that happens, all right? So after death, you're clearly not respiring and you're not making ATP. So if you lose ATP, there's 2 things that will be prevented. So first of all, you won't be able to pump calcium back into the sarcoplasmic reticulum, meaning that there will be lots of calcium in the sarcomere, and the binding site for the myosin on the actin will be revealed, so the head groups can interact. And then remember, for detachment, we also need ATP. So if we don't have any ATP, the head groups will remain attached to the actin and keep the muscle contracted. So therefore, this is why you have that stiffening of the body after death. Eventually, the proteins will start to break down, right? And then they will release. And then there's a certain amount of time that that's required. And I bring it up in case any of you are in criminal justice. So That's how they consider time death, depending on, what you're dealing with. Obviously, if you still have, you're not skeletal remains, right? But if you have a body there, you can sort of time the death based upon whether it's stiff or loose again. Okay. Now, there's a few other proteins that are required to keep the sarcomere together. So one of them is called titin. And titin, it runs through the core of, here you can see it. It runs through the core, this is it here, it's a springy protein that runs through the core of the myosin, okay? And what it does is it helps anchor, so it runs through, see here, it's the core of the myosin, and then it attaches to the Z disc. So it helps to stabilize the position of that thick filament, right? You see how it's attaching here, right? So it runs through the core and it binds to the Z disc, all right? And it also, you can see it, because it runs through the core of the thin filament as well, all right? So that then keeps everything aligned, all right? So titin, a huge springy protein, runs through the core of the thin filament, anchors it to the Z disc and the M line, all right? And it helps to stabilize the positioning of the thick filament, all right? And then it also helps prevent overstretching, right? So that's the other thing that it does. So that's titin. Now, now the next one is nebulin, all right? So nebulin aligns through the thin filaments, okay? But it does not attach to the M line, all right? So it attaches to the Z disc, but not to the M line. So here you can see it here, it's in blue, all right? And here you can see the titin, right? So this one sort of shows you. So it's sort of, you've got the nebulin running through the middle here, and as well as titin, and then titin is going to extend out and also align with the M line. I mean, with the thick filament, all right? So you can sort of see how it's, how it sort of aligns to this, right? So it attaches to the Z disc, but does not extend to the M line, right? You can see that it's, going through here, but it doesn't really attach, right? So it's kind of a weird drawing, all right? But it's really important for the elasticity of this, right? So never get perfect drawings. This one is a better drawing, all right? So here you can see it's sort of misleading that it sort of doesn't quite attach to this M line, but it does go through the core, all right? It goes through the core of the thin filament, attaching to nebulin, and attaches to the thick filament to help attach it to the Z disc, all right? But it's not attaching it to the M line, all right? Just so that you understand the difference there. Okay, now we have another protein called dystrophin. And dystrophin is, if you know about muscular dystrophy, so it's clinically important, all right? So here you can see dystrophin. It's attaching to the thin filaments, all right? And what it's doing is that it's sort of aligning that actin in the outer parts of the myofibril to the actual sarcolemma through another protein. There's some linking proteins here. And here you can see the sarcolemma. And then remember, we have the endomysium on the outside of that, all right? So this sort of anchors the cell, including the filaments, to the endomysium, so that this is all aligned perfectly, all right? Now, if we have a breakdown of that, right, there's a genetic defect, it's an autoimmune breakdown of dystrophin, that then causes this muscular dystrophy, or the muscle weakening, all right? All right. Now, motor unit. So a motor unit is one motor neuron, all right? And all of the muscle fibers are the muscle cells that it controls, all right? So you can have different numbers of muscle fibers associated with different motor units. But the thing to understand here is that when you have this, and I'm going to refer to my notes here. Okay, I'm just looking at page 4. What did I write for titin and dystrophin and nebulin? Let me just go back. I want to make sure I said everything that's in my notes. So I said titin Elastic stabilize and anchor myosin thick filaments. Dystrophin attaches the myofilaments to the sarcolemma. Nebulin allows for actin thin filaments to wind and create a helix, keeping the actin aligned. All right, that's what I said. All right. Now, motor units. All right, so here you can have large motor units and small motor units. So small ones, they usually 3 to 6 muscle cells per neuron. And this would allow for very fine movements, but less force. And then you can have large motor units, which have maybe thousands or hundreds of cells attached, associated with a neuron. And these have power, but not very much finesse, we'll put it that way, okay? So the amount of muscle tension then that's provided by the motor unit is going to depend on the number and size of the muscle fibers in the unit. And overall, so the other thing that you should notice here is that the fibers that are associated with one particular unit can be throughout the muscle, all right? So the number of motor units active at any one time in any particular muscle will also depend on the full muscle tension, as will the frequency of stimulation. We'll talk a little bit more about that, all right? So here you have your difference between small and large motor units, right? So small motor units, fine degree of control, but not much strength. And large motor units, lots more strength than they do control, all right? Now, let's think about how this is happening, all right? So now we're talking about the neuromuscular junction and exactly how that electrical potential is going to spread across the sarcolemma and then down into those T-tubules, all right? So later on in the semester, you'll learn about neurons, all right, and how the action potential is carried through there and how they release neurotransmitter. But I'll sort of give you a little bit of a heads up on that. So basically what's happening is that your electrical potential is going to travel down the axon to the terminus. And that is actually going to open up calcium channels. So we call them voltage-gated calcium channels, all right? Because they open in response to a change in charge, all right? And those calcium channels are going to stimulate the vesicles that contain neurotransmitter to fuse with the terminal end ball, all right, the axon terminus, and release the neurotransmitter into the junction. The neurotransmitter then, and when we're talking about skeletal muscles, is called acetylcholine. And it then diffuses across the junction and binds to its receptors on the muscle cell. And here you can see on the muscle fiber, the sarcolemma here, you can see these invaginations. This is increasing the surface area. So you can have lots and lots of these receptors here, okay? So, so the muscle cell then can have millions of these acetylcholine receptors, all right? And these folds are going to increase the number that we have here. Now, the other thing to think about is that here we're talking about this basal lamina, all right? So here you've got this basal lamina that's sort of around here. So this basal lamina is a thin layer of collagen, all right, that's going to separate when you're going to talk about Schwann cells later on, okay? So Schwann cells, when you talk about neurons, are insulation for the neuron and the muscle cell, all right, from the surrounding tissue, because what you're doing is you're sort of isolating the electrical potential. So sometimes there would be, in certain cases, if it's a myelinated muscle fiber, there would be a Schwann cell wrapped around here, and you'll learn about that later, okay? Now, how do we get rid of the neurotransmitter when we want to turn this off? Well, there's an enzyme here called acetylcholinesterase, which will break down the neurotransmitter. There's also some diffusion of the neurotransmitter out of the synaptic cleft, all right, at this neuromuscular junction. That can also happen despite this basal lamina, all right? Okay, now, so here we have these voltage-gated calcium channels in the neuron. That allows for the fusing of the neurotransmitter-containing vesicles, and the neurotransmitter, shown in sort of red here, binds to its receptor on the membrane of the muscle cell, all right? So this is synaptic end plate, synaptic end bulb, and this part here where we have all of these receptors is called the motor end plate of the muscle cell. And then what's going to happen is that's going to bind to these neurotransmitters. All right. Now this then is going to be used to set up a action potential in the muscle cell. So let's talk about that. What is an action potential? Now I want you to think back, right? So when we talked about cells, I said to you that every cell membrane has a charge across it. All right. And it's only muscle cells and neurons that can change that that potential or that charge gradient across that that membrane. All right. So if you look on page 6, I kind of drew this out. I said, you know, we've got sodium and potassium, right? And remember, I had said to you that that potential that all cells have across their cell membrane is established by the sodium-potassium pump. Okay. And we needed that to keep the gradient so that things will keep working for homeostasis. And I had said to you this is going to be really important when we do muscle and nerve. So let's talk about that, right? So it works out that outside the cell, there's a higher concentration of sodium than there is inside. And inside the cell, there's more potassium relative to the outside, right? This is why the pump is moving these things in opposite directions against their concentration gradient. Now, what's going to happen is that when the cell flips its potential, what's going to happen is that the charge across the membrane is going to decrease, rise, all right? So I want you to see the difference here is decreasing, all right? So we have this thing. So it's basically, here they're talking about the membrane potential to be about minus 70 millivolts. But practically speaking, most of the time it's about minus 90 millivolts, and that's what you'll see. And what will happen when the action potential is stimulated, that the charge is going to come closer and closer to what we call a threshold, all right? So here, figuratively speaking, it's set at about 0, okay? So it's going to sort of keep rising, and then eventually it's going to become positive. And then what's going to happen is that when the action potential is done, this is going to repolarize, and it's going to come back down, back to where it was, and the sodium-potassium pump is going to affect that. So now what's going to happen is that as sodium comes in, you're going to depolarize, and as potassium goes out, you're going to repolarize. And in fact, you could also hyperpolarize the membrane if you allow even more potassium to go out, which is going to be more important when we talk about what happens in neurons, all right? So think about that. So remember, you have more sodium outside and more potassium inside. We've got a negative charge, typically about negative 90 millivolts across the membrane. If sodium comes in, this is going to depolarize. And if potassium goes out, it's going to repolarize or even hyperpolarize. All right. So when we're talking about an action potential in a muscle, we say that the resting potential is about negative 70 or maybe even negative 90 millivolts. All right. And what's going to happen here is that when the neurotransmitter binds to its receptor, it's going to open, it's actually a potassium, a voltage gated or a chemically gated sodium channel. So chemically gated because it's going to be opened due to the neurotransmitter. And that's going to allow some sodium to come in. And when it does that, it's going to get it closer and closer to what we call threshold. All right. So in this diagram, threshold is about minus 55, because remember, as sodium comes in, it's going to go down its concentration gradient, and it's going to bring that membrane potential upwards. Now, if it hits this, what we call a threshold potential, what will happen is voltage-gated sodium channels will open, and sodium is going to rush in. Now, these voltage-gated sodium channels will close at a certain voltage, and it tends to be around plus 30 millivolts. And at this plus 30, this is when the potassium channels, these are now voltage-gated potassium channels, become fully open. They actually open a little bit before that, but they become fully open at about plus 30. And then what's going to happen is that potassium is going to go down its concentration gradient and flow out of the muscle cell. And that's going to repolarize the membrane. Okay? So you can see here in orange the concentration of sodium, and in green the concentration of potassium. And then in blue, the actual charge across the membrane. All right? All right, so now excitation, let's go through some terms here. Excitation is a process which the nerve action potential leads to a muscle action potential. So that means excitation. Excitation, contraction, coupling, when you see that, that refers to how we link that action potential to what's happening in the sarcomere, right? So the activation of the myofilaments and allowing them to contract. Then contraction is when the muscle fiber develops tension because the sarcomeres are shortening. And relaxation is when that stimulation ends and the fibers then release and they recoil back to their original length. Remember, I said to you, the action part of muscle is contraction, and the resting part is when it all relaxes and extends back out to its original shape or length. Now, we can measure these voltages using probes, right? So we can measure the actual charges in the charge potential across the membrane in the neuron and the charge potential across the membrane of the muscle fiber. And obviously, the neuron has to depolarize before the muscle fiber, and then the muscle fiber is going to depolarize, which is showing you here. So we have the neuron action potential, we have the muscle fiber action potential, and this is all going to precede the actual contraction of the muscle. So if you would look at the muscle tension as a graph, you would see this part here is the latent period, is when this electrical stuff is happening, right? So this is the delay to the actual contraction from the movement of the electrochemical potential. Then we have a contraction phase where the muscle will shorten, and then a relaxation phase where it will recoil. Right, so recoil when the head groups detach from the actin and everything goes back to its original length. Now, one of these contraction and relaxation phases, this is what we call a muscle twitch. Okay, so a cycle of contraction and relaxation is a twitch. And we call it all or none. So either the muscle fiber is going to contract or it won't, all right? So once it contracts, and, but you need to get to a certain potential in order for it to contract, all right? So that goes back to how much of a depolarization is happening, and that will depend on how many neurotransmitters are binding to the receptors and how many of the chemical activated gates are opened due in response to that neurotransmitter. Now, if we look at this, all right, we can summate these excitations, all right? So if you're looking at muscle force, all right, and you've got these action potentials coming through, and a fiber, all right, can twitch, right, can relax, contract and then relax. And then it can contract and relax again, all right? Now, if it is stimulated before it has a chance to fully relax again, right, as it can then add up or summate those potentials. And the more times it's stimulated and the less time that the muscle itself has to relax with all of the motor units that are being stimulated, we can reach a point where we have a full tetanus, or a muscle that remains contracted because the muscle fiber remains contracted. All right, so if we look at this in this way, so we can have these twitches. So low frequency stimuli, we say, produce these little, twitches, right? And the fiber will contract. all right? But that doesn't mean that the whole muscle is going to be fully contract. Then we can have higher frequency of these, and we call that temporal summation, because in time you get more and more and more of these twitches. And as we do, the tension is going to rise. All right, so we say it's like a wave summation, because we have a temporal wave where more and more of these, fibers are going to be activated, and they'll have less and less time overall for the muscle to relax. All right, and then we can have a full-on tetanus or a fused tetanus, and typically this happens for a particular muscle. We call it fused tetanus. It's actually kind of unusual. All right, for it to be full-on fused tetanus. Usually what's happening is that motor units are going to take turns in their contraction, and that's going to dictate how much of the muscle remains contracted. And you can, and the brain will sort of regulate how much tension there is in a muscle, dependent on how many motor units are contracted at any one time. But very rarely do we have all of the motor units completely contracted, all right? And that's what we get, fused tetanus, all right? So if we look at this, we can have a spatial summation, right? So on a muscle fiber, right? So if we have a spatial summation and a whole, within a certain region, a certain number of neurotransmitters binding, we can then, because remember, you're talking about a whole very long muscle fiber, right? You're talking about a cell that, I think I used the sartorius muscle as an example, right? From the outside of the hip to the inner part of the knee, right? So, spatially, if you have a whole bunch of motor end plates activated, that's going to cause a spatial summation within a certain region. But also, if many of them are activated with time, that's like a temporal summation or a wave summation, all right? And as you summate, you're going to have more and more of these motor units that are activated. So here you can see on this picture the number of muscle fibers that are motor units that are activated by how many muscle fibers are involved. And then eventually you can reach maximal contraction, all right? And you can see that some of the motor units are probably going to relax in this, but if you've got enough, you're going to have maximal muscle contraction. But if all of them are contracted, all right, you're going to have full-on tetanus, all right? Now, tetanus, that is a problem. You can also call tetanus as a bacterial infection, which is why you get vaccinated. I just got vaccinated for tetanus, again, as a stipulation for the job I'm going to be starting. Basically here you have a vaccine that is against this toxin, so that if you get a puncture wound and you get this bacteria, this toxin won't cause death, right? So this toxin will overstimulate the motor neurons and you get fused tetanus. And the reason this is deadly is because your diaphragm, remember your diaphragm muscle, right, between your thoracic and your abdominal cavity, it's actually skeletal muscle. So if it's fully contracted, you won't be able to breathe. So you have someone can actually be asphyxiated because of that. Now, there's this difference between the types of contractions we can have. So we can have isometric contractions, which are shown here. So that is no movement, but clearly you can see that the muscles must be contracting. So these motor units are taking turns in this isometric contraction to sustain holding a book outright, right? So holding a book steady. So that is an isometric contraction. You don't actually see movement, but you know that the muscles are contracting. Then you can have isotonic contractions, right? So there's isotonic, can be concentric or eccentric, all right? So if they're concentric, what you have is a situation where your muscle is shortening, all right, as it maintains tension. So that would be like a flexion. And then eccentric, all right, is when the muscle is going to lengthen as it maintains tension. So this is the overall muscle, all right, not individual units, all right? So the overall muscle is going to lengthen, all right? So these are isotonic. All right, now, getting into energy supply. Now, if you're talking about muscle, clearly you need energy, right? You need ATP. So how does the muscle cell do this? Well, we talked about myoglobin already, right? We said, I told you that myoglobin is a way that the muscle cell can store up some oxygen so it can make ATP as needed, all right? So when we look at this, there's two ways that the muscle can make ATP. So obviously aerobically, right? So that's using the mitochondria. And remember, when we do that, we make lots of ATP. The other one would be anaerobic respiration, which is called fermentation. Allows the production of some ATP in the absence of oxygen. That's what anaerobic means without oxygen. It doesn't produce very much ATP, but it produces a lot of lactate, right, which we need to get rid of, right? So that causes muscle pain, all right? So when we're looking at this, the way that ATP is going to be produced is going to depend on the oxygen supply, all right? And whether you're going to use as an energy source glucose or fat, all right? So if we look at this, and we've got my little diagram on the top here, and I think this is from a Saladin text, right? So there can be some aerobic respiration using oxygen from myoglobin, all right? So stored up oxygen, so we can have a little bit of aerobic respiration for that. We can also use some of the ATP that's already in the cell. Then the next thing that the cell will do is try to recycle ATP using something called the phosphagen system. Basically, it's going to take phosphates off of other stuff and put it back onto ADP. All right, so we'll talk a little bit about that. Then, if the cell runs out of oxygen, it's going to start fermenting. It's going to use anaerobic respiration or basically glycolysis, and it's going to use and it won't use oxygen in this place, but will make a little bit of ATP. And then it will use aerobic respiration, if possible, all right, to produce lots of ATP aerobically using oxygen, right? And then, of course, then we're tired. We're tired out. Once we exhaust this, we're tired out. So let's go through these ideas. All right. So energy for muscle contraction. So first of all, ATP in the cytoplasm, or using some aerobic respiration using stored up oxygen in myoglobin. Then it will try to recycle that ATP by taking phosphates off of creatine phosphate, which is a molecule that we'll get into a little bit of detail in the next slide, or from ADP, making AMP. Then it will try to use ATP using glycolysis, anaerobic respiration, or without oxygen, and make a little bit of ATP. Or it's going to make ATP aerobically using mitochondria, right? And remember, when you do aerobic respiration using mitochondria, you can feed in from glucose, you can come in through fat or from amino acids or protein breakdown. All of it will feed into this. All right, but usually we learn about it using sugar, all right? And that will be making ATP in the presence of oxygen, all right? So here's, all right, so let's talk about this phosphagen system, because that's kind of weird, right? So basically what will happen is that the cell will try to recycle ATP using phosphates from other molecules. So one way it will do that is it will take ADP, because there's still a phosphate there, and rip that phosphate off and add it back to ATP. So it will sort of to other ADP molecules to resynthesize some ATP. And the enzyme used to do this is called myokinase. Also within muscle, there is this molecule called creatine phosphate, or creatine phosphate. And there's an enzyme called creatine kinase, which will take the phosphate off of that. A kinase is always a phosphorylating protein enzyme. And it will take that phosphate and phosphorylate ADP to make ATP. All right. And you can see the amount of energy or ATP that you're going to get from doing that. It's about 6 seconds worth of it. So not very long, but a little bit. Okay. So if we look at this, right, so we say using the amount of ATP that's already in there, or using myoglobin to make aerobic ATP will last you about 3 seconds. Recycling would be using phosphocreatine or ADP, maybe about 6 seconds to 30 seconds. Using anaerobic respiration, that will get you to about 30 seconds. And then opposed aerobic respiration using oxygen from the bloodstream, that can sustain you for a long time, right? Especially if you're trained. We'll talk a little bit about training. That can sustain you for hours if you're in the right headspace and you're well trained. All right. Okay. Let's see where we're at here. Okay, so we're now on page 8 of my notes. All right, so skeletal muscle fiber types. We've got 2 types. So they're not all alike in appearance and function. So you probably know this if you've eaten meat, right? So some meat is red, some is white, and some is in between. So why is that? Well, we've got red muscle. That is what we call slow twitch fibers. Red muscle or slow twitch fibers has, and this will get like the dark meat in chicken, right, or turkey, has a low glycogen content. And we're looking at page 8. I have compare and contrast, slow and fast twitch. So we have a low glycogen content, but a high myoglobin content, right? So myoglobin, I pictured it here. So myoglobin is similar to hemoglobin in that there's an iron there and it binds oxygen. And that gives it a red color, right? So if you it's a rust, right? It gives it a red color. So that's why it gets, sort of reddish, right? So that's why it's red muscle, right? There's a high myoglobin content. Also has lots of mitochondria. So that means it's really good at making oxygen, ATP with oxygen, right? And a really good blood supply, which is the other reason it looks red, because then you're going to have the hemoglobin in there as well, right? So that's red muscle. We call it slow twitch fibers, all right? So this is really good at aerobic metabolism, all right? You can take that glycogen, take the sugar out of there, and then sort of feed in through glycolysis to the mitochondria, Krebs cycle, and oxidative phosphorylation, and we'll make lots and lots of ATP. Then we have white muscle, which we call fast twitch. So this is different, right? This is like the white meat and chicken breasts. Here you've got a storage of glycogen, much more than you have with the slow twitch. You've got lots of glycogen stored up. But you have less myoglobin and mitochondria and blood supply. So you don't have the ability really to use a lot of oxygen. And therefore, you've got anaerobic metabolism, all right? And if you look at this, these fast twitch and slow twitch, so red and white fibers, are sort of mixed in muscle. So here you can see these different fibers, and you can see the different colors of them. If you look at them under the microscope, it's really quite cool. Not that they're red and pink, red and white, but you can see that there's a mix. And here you can see that you can actually have a mix of those fibers, and you can get somewhere in between if you're looking at meat, all right? So let's think, slow twitch, we say that contracts slowly, and you use the slow twitch fibers for endurance activities. So when you're going through aerobic training, what you're doing, or endurance training, the body is going to learn how to use the oxygen much more efficiently. And with time, you will increase the number of capillaries, your myoglobin content, and the number of mitochondria in those muscles, all right? So this is really good if you're a marathon runner, right? So that's for your aerobic training. On the other hand, slow twitch, all right? So slow twitch versus fast twitch. So fast twitch fibers, those are going to contract very quickly, and they're used for brief, high-intense activities. So like, strength training. So here the muscles learn to work harder and quicker. You're going to build more myofibrils in your muscle fibers. You don't actually increase the number of muscle fibers. You're going to make those myofibers bigger because you're going to make more myofibrils. And you're also going to gain more glycogen and more creatine phosphate for quick energy, all right? So quick boosts of energy. So that's the difference between the slow twitch and the fast twitch. So let's talk about what happens after exercise exhaustion. So there's a number of things that are going to happen, right? So on page 8 of my notes, I go high intensity and low intensity. So high intensity, short duration exercise. So that would be like here, fast twitch work. Potassium accumulation in the T-tubules. Excess ADP is going to slow down the movement and calcium release, all right? And then for high intensity, so this is endurance work, fuel depletions, you're going to lose glycogen and glucose. Loss of electrolytes, so because of that, then it's not going to work very well anymore, and you're going to lose them through your sweat. You're also going to have like fatigue. The brain is just going to be tired, not only of getting through that wall of endurance, but also just from sending the signals from the brain to the muscles, all right? So here on this slide, you can see like a whole bunch of different things that can happen. Increased need for kidney clearance of waste products. So what is that? Well, that's your lactate, right? So if you're doing anaerobic respiration. Depletion of glycogen. If you deplete your liver of glycogen, you're going to lose glucose in the blood, right? Rapid exchange of gases in the lungs. It's just going to get tired. Altered electrolyte balance. Nervous system fatigue. Depleted triglyceride stores. So you're going to lose fat. Rapidly increased heart rate. So that's going to tire you out as well. Microtraumas. You're going to get damage to the muscle fibers themselves. And then, fatigue in the peripheral nervous system. So then what we have to do, and we have to restore, there's got to be some period of time to restore that. And we call this the oxygen debt, right? So that goes by this EPOC. So that's excess post-exercise oxygen consumption. So after exercise, and so you can actually increase the basal rate like 6 times of oxygen usage to replenish ATP, replenish the myoglobin stores, right? So add oxygen back to that. Remove the lactate. So that's going to be done in the liver, and that's going to need oxygen. And then restore your tissues, and that is also going to use oxygen. So this is sort of the debt that you have to pay after working out, all right? And that, and depending how well trained you are, that can take different amounts of time. All right, now the last little bit is comparing skeletal muscle, like we were just discussing, with cardiac and smooth or visceral muscle. All right, so remember my chart, right? So skeletal muscle, before we talked about, this is when we did tissues, which ones are striated, which now means that they have sarcomere structures. So both skeletal and cardiac are striated. But smooth muscle is not striated, right? So it doesn't have a sarcomere structure. It does have actin and myosin, but you'll see, I'll show you it's in a different arrangement. Skeletal muscle is voluntary, but cardiac and smooth are involuntary. So now let's look at a little bit more detail. So cardiac muscle fibers versus skeletal muscle fibers. So how are they similar? Well, they're striated, which means that they have a sarcomere structure. But they're unlike that unlike skeletal muscle, because the muscle fibers are shorter. They've only got one nucleus. They may be branched, and they're involuntary, okay? Now, another major feature that cardiac muscle has that skeletal muscle doesn't are these structures here that are shown in these dark bands, and those things are called intercalated discs. These intercalated discs have tight adhesion junctions to keep the muscle from tearing, because you don't want to tear your cardiac muscle. That would be crazy. But you also have gap junctions there. Now, those gap junctions are really important. Remember when we talked about gap junctions, that allowed for the movement of molecules and ions, if they're small enough, and ions directly from one cell to the next, all right? So here we can see these intercalated discs. You can see that they're at the junctions between these cardiac muscle fibers. And basically what this means is that the stimulus can move directly from one muscle fiber, or the action potential can move directly from one muscle fiber to the next. It doesn't require its own innervation to do so. And this is really important with the coordination of the muscle contraction, or the cardiac muscle contraction, as you'll see when you do heart in semester 2, all right? Now, the other thing that cardiac muscle has that skeletal muscle does not are pacemaker cells. And you probably know that there's pacemakers in the heart. There's 2 of them. The main one is in the SA node, and the other one is in the AV node. And I've got this video here. You can sort of see what happens with these pacemaker cells. But basically, they have a drifting resting membrane potential. The resting membrane potential gradually comes to threshold and fires on its own. And it sort of sets the pace for the heart. And the spread of ions through those intercalated discs to the surrounding muscle fibers can then allow for that muscle contraction to spread from the pacemaker, where the pacemaker cells are, into the other muscle fibers, or other cardiac muscle fibers. The brain can influence this to a certain extent, but understand that the pacemakers will beat regardless. So if you, say, get a heart transplant, the heart is going to be influenced by its own pacemaker. You're not going to get that innervation into the transplanted heart for a long, long time. It can be regulated using hormones. But suffice to say, this is the heart can beat on its own. That's why it has its pacemakers. Now, smooth muscle fibers versus cardiac muscle fibers, all right? So like cardiac muscle fibers, smooth muscle fibers are involuntary. They are electrically, some of them can be electrically linked to each other using gap junctions, and some will have pacemaker potentials. Now, this is going to be dependent on where they're at, okay? Here, the pacemakers are under control of the brain. The other thing is that typically smooth muscle comes in layers in certain places. So around the respiratory system, the digestive system, and the circulatory system, we've got this ring of smooth muscle. And definitely around the digestive and the respiratory, we've got two layers. And those two layers of smooth muscle are at right angles to each other. So they allow for contracting and shortening of those tubes, so the digestive system and the respiratory system, all right? So and especially in the digestive system. In fact, in the digestive system, in the heart, you have, not the heart, sorry, in the stomach. In the stomach, you have a third layer where it's sort of at an oblique angle. And that can allow for mixing. And you'll learn about this in semester 2 when you do digestive system. Now, unlike cardiac muscle, they don't have sarcomere structure. So unlike cardiac and skeletal muscle, there's no sarcomere structure. And they do not have intercalated discs, even though some of them have pacemakers, all right? Now, so what happens here? So since there's no sarcomere structure, we still have myosin and actin, but it's arranged differently. So first of all, the head groups on the myosin will walk in both directions along the actin. So this causes a weird shrinking of the cells, all right? So that the filaments don't slide past each other. They actually shrink the cell. So the whole thing sort of scrunches, all right? So you have this sort of situation where they're scrunched, and then there's these filaments that connect the cells in the whole network of smooth muscle, so that they'll sort of scrunch and pull on the next cell as well. All right, so this is sort of have like a chicken wire arrangement of these filaments. So if you look at them, there's this connective tissue between this as well. So here you can sort of see that the myosin is still in the middle, and then the actin is sort of around the outside, and it sort of allows for this sort of shrinking. And then there's intermediate filaments, if you remember your cytoskeleton, that sort of connect these nodes where the actin is attached to, all right? And then you've got these connections, these nodes are called dense bodies. And then you've got these connective tissue on the outside that connects the dense bodies from one cell to the next and allows for these things to contract together depending on the arrangement of the motor units. So here you can see a relaxed versus a contracted cell. It contracts in 3 dimensions, all right? So when I was talking about number of motor units, you can have single unit smooth muscles, which are going to be connected using gap junctions. So this is where you want them all to contract together. So you can imagine here, this is the gut, and you've got your two layers, your circular and then your right angles longitudinal smooth muscle. And you're going to want these to sort of contract together as a single unit. All right. And there's these swellings where the neurotransmitter is released from the neurons called varicosities. All right, so and then it comes down here, and then some of the cells have the receptors, and then they pass the information on to the other cells using the gap junctions. Then you can have multi-unit smooth muscle cells, which are not electrically linked. So this is when you have a bit more fine control. So this would be like the smooth muscle that controls the flexing and the relaxation of the lens in the eye, for example. So these ones are going to have their own receptors, and they're not electrically linked using gap junctions. Now, this last thing is just to sort of show you the differences in smooth muscle, because you've got all kinds of different types of smooth muscle in the context that you have. So you can have, say, and what happens with the contractual force that you have. So you can have like a phasic smooth muscle, which is usually relaxed, which would be like the esophagus, and it will contract when you swallow something. Right, so you have this contraction force, and it will move down the esophagus. Then you can have like a phasic that moves in cycles, which would be like the intestine, right? So it will, if you have peristalsis. And in fact, when I used to work in the lab with gut, I was studying colon cancer. And when I removed the intestine from the mouse that I was studying and just put it in a buffer, the small intestine or the large intestine would continually contract, even though it was no longer connected to the animal. So there's a pacemaker there as well, and you can see that it would have this sort of like a phasic smooth muscle that cycles. Then you can have like a tonic smooth muscle. So tonic meaning constantly contracted, which would be like a sphincter. So in this case, it's going to be constantly contracted, and then it's going to release when it opens, and that's what that's showing there. And then you can have a smooth muscle that's a tonic as well, so there's going to be always some tension, but its variation is going to be changed as needed. So that would be like what you have around your blood vessels, so vascular smooth muscle. All right, just to think about, because there's different contexts in which you have smooth muscle. And I think that's it. That goes through all the learning outcomes. And there's a lot there. So that's why it took a few days to go over all of this. But I hope this has helped. And this will be my last video for you. So I really hope, I'm not going to start crying now, but I really hope that you have a good semester. And yeah, and hopefully things will go well as you go through the rest of the course. All right, thanks so much for being my students, and I really have enjoyed teaching you.